All right, well, it's 6 o'clock, so let's just go ahead and jump on in. I'm excited for our conversations. So first off, howdy and welcome everyone. Thanks for being here on a Friday night. We appreciate you guys being here. And we're really excited to introduce A-STEP's annual town hall event. So normally we do try and have this conversation in person, but this year we're excited to be hosting for you via Zoom. We have a lovely speakers already set up for you guys. And the moderator of tonight will be Dr. Barbara Gastel. And so we're so excited to have you all here. We can't wait to, see these presentations together and join in a community discussion. So we hope you enjoy the resources within science talk tonight. All right, Dr. Gastel, take it away.
Thank you very much. As mentioned, I'm Barbara Gastel, and I'm pleased to welcome you to this A-STEP Town Hall titled Resources Within Science, Addressing Science Skepticism with A&M Experts. First, let me thank Aggies in Science, Technology, and Engineering Policy, better known as A-STEP, for organizing this event. I also want to thank the Scientific Research Honor Society, Sigma Xi, and the Office of Graduate and Professional Studies, or OGAPS, for their support of this event. And thank you for asking me to be as moderator. By way of background, I'm a professor in the College of Veterinary Medicine and Biomedical Sciences, and also at the College of Medicine. And I coordinate the graduate program in science and technology journalism. And so I have really broad interests in science, technology, and medicine, and the communication and their place in society. And so I'm especially looking forward to this evening's program. The plans for this evening are as follows. I'm going to make brief introductions of all four speakers. Then we'll have two of the presentations and a Q&A and a brief intermission. Then I will reintroduce speakers for people who weren't here, have the other two presentations and more Q&A. And then I will turn this back over to the A-STEP leadership for some closing items and some open discussion. So in terms of our four speakers, I think we have a wonderful panel, and I could probably take up the whole first hour listing all their honors, but I won't do that. I will limit the introduction to a very few sentences per person. Our first speaker, Dr. Stephanie Payne, is professor of industrial and organizational psychology in the Department of Psychological and Brain Sciences. She also has an appointment in the Mays Business School and is a faculty fellow of the Mary Kay O'Connor Process Safety Center at Texas A&M. Her research interests include safety climate, individual differences, performance appraisal and management, and work-life balance initiatives. Our second speaker, Dr. Keerti Rathore, is professor in the Department of Soil and Crop Sciences, and he has established Texas A&M as the premier academic institution in the area of cotton improvement through genetic modifications. Like our other speakers, he has an impressive publication record, and he is also active internationally. Our third speaker, Dr. Charles Long, Chuck Long, is professor in the Department of Veterinary Physiology and Pharmacology, and he has conducted research in industry, government, and academia. His laboratory is currently working on a number of projects involving genetic engineering in cattle, goats, sheep, and horses, and his projects have many implications for human and animal health and for the food supply. And our final speaker, Mark Holtzapple, is professor of chemical engineering. He has had wide research interests. Some of them are bio-based fuels and chemicals, food and feed processing, water desalination, air conditioning, high efficiency engines, jet engines, and vertical lift aircraft. He's developed a wide variety of technologies, and received multiple awards. And I guess I'm most used to seeing him in his role as a horn player in the Brazos Valley Symphony Orchestra. So, that is our introduction. And so I think without taking further time from the presentations, let me turn this over to our first presenter, who I believe will be Dr. Payne.
Okay, there you go. I was muted. Okay, you guys can hear me. You can see my screen, right? Are we good? Yeah. Okay. So
yes, we're good.
Perfect. Okay, very good. Well, I have to admit that I'm not well versed on the skepticism in science per se, but I approached this topic. Was happy to be invited, and I approached the topic from kind of maybe a bigger bird's eye view, but also recognizing my role in psychology. So I hope this isn't too basic, but I think it might be some fundamental kind of core concepts that we may circle around a little bit tonight, depending on, I don't have a full vision of what exactly we're going to be doing in the grand scheme of things here. But so when I was asked about talking about skepticism in science, I did do a little bit of research, and I thought it was interesting that when you look at what do we know about scientists, turns out that scientists tend to have, certain kind of personalities and approaches to life and perspectives. And one of the books I came across talked about a scientific attitude as having these three characteristics: curiosity, just a desire to, answer questions; skepticism, ironically, right, by virtue of just being scientists, we ourselves tend to be, curious and also skeptical, asking lots of questions and wanting to get to the bottom of the truth, right? And then also humility, which I think is also an important aspect here. We recognize we are human. We also recognize that there are limitations to our science, and always looking for ways to improve it. So I just wanted to mention that because I think it helps to kind of put things into the bigger perspective. I know that generally when we're talking about skepticism in science, it's coming at us, if we're the scientists from the public, people who maybe don't necessarily identify themselves as scientists. Now I'd like to kind of just remind us of what is science. And I borrowed from my research methods classes where we talk about psychology as a science, which isn't always necessarily given the respect that I think it deserves as a science. And just really quickly remind us, well, what do we know about science? What is science? Well, generally it's usually defined with regard to these two primary characteristics, being empirical and being objective, right? And I'm sure this audience is very knowledgeable about what this means, but again, just as a reminder, empirical meaning that we rely on experience and observations on data, and that we ideally strive for an objective view on these data so that everyone is perceiving it the same way, or analyzing and gathering the information the same way, we could replicate it and so forth. And so what is beautiful about science is that it allows us to test for superiority. So a lot of times in psychology, we find ourselves in a situation where people feel like what we talk about may be common sense, but the reality is that there's still surprises out there in terms of what we know about human behavior, and science allows us to determine, more in a scientific way, what is the truth and what goes beyond necessarily common sense. A little bit more about the characteristics of science that make it superior and better than any other way of knowing, is this list of kind of characteristics of science. So the first and foremost, the most important thing is control. And basically what control does is it allows us to rule out alternative explanations. And it's the primary thing. That's why I put it in with extra stars there, is it's the thing that makes a science so special in some ways. Other characteristics are that it's tentative, right? We learn things. Sometimes we get them right. Sometimes we don't necessarily get them perfect. And we continuously add to our knowledge. So it's not just, what we learned today, end of story. We continue to add to it. So it's just to what we know today. And then we make corrections. So related to that, so self-correcting. In science, generally, we're looking for replication to kind of to bolster our the robustness of our research. And so we look to as part of the method, the scientific method, right, is to have that replication. And so sometimes that means that we find new things and we correct what we previously thought before then. A few other things, so it's progressive, right? So we're constantly accumulating more, which sometimes makes it a little bit intimidating as you approach a new topic area. It's also theory driven, such that we're interested in the questions of why and how, and this helps us to guide our research as well. We oftentimes start with theories and then look for the data, but sometimes we work the other direction as well. I already mentioned it being both objective and empirical, again, relying on data and striving for objective views on the data. So it's not just my personal perspective that people could replicate, get the same results because they would gather the data the same way. And ultimately, it's also parsimonious in that we look for the simplest explanations for the most part. All right, so a final just kind of note about science, and then I want to talk a little bit more about psychology as science. There oftentimes I present 3 goals of science, but I've since added a 4th. And so you think about the big picture, what are we striving for in science? We have description. It's often the kind of early phases when we're first kind of trying to understand a given phenomenon. We might just be at that surface level where we're describing and we're looking for patterns. That's what we sometimes call laws, as well as ultimately causes. The second goal we're striving for is explanation. So we want to go beyond describing. Now we want to explain. And then in psychology, we're all about studying human behavior, also animal behavior, but trying to explain that behavior. And once we can explain it, we can take it one step further. We can start to go down the path of prediction. And ideally, this lends itself to, once we can start to make predictions about people's behavior, then we can start to move in the direction of facilitating both prevention as well as promotion. And that just kind of gets into the 4th one that I've added there is ultimately changing potentially behavior. So a lot of times in psychology, we're looking to improve or increase certain types of behaviors and decrease or prevent other types of behaviors. So big picture, kind of reminder of kind of what are we ultimately striving for when it comes to science. How am I doing on time? A few more minutes, are we?
Yes, you have, yeah, a few more minutes,
okay.
About halfway through, maybe about 7, 8 more minutes. Something.
Okay, fine. I'm happy to have you just cut me off. So the last 2 slides I have are just kind of some anecdotes of things that as I think about doing good science and psychology, some things that make it a little bit challenging. And I think that ultimately sometimes contribute to the skepticism that does get created by others outside of psychology. So the first thing is human behavior is complex. And so understanding people's behavior, turns out it's not as simple as just one independent variable or one cause. There's lots of things that contribute to human behavior. And so that makes it complicated, right? It's not a simple phenomenon. Doing good science takes time. And many of the things that I've listed here actually kind of speak to that issue of taking time. A lot of times we work in environments that are somewhat bureaucratic, and so we have lots of steps and hoops to go through in order to get our science accomplished. I think about working in an academic institution and various reporting that has to be done and steps that are involved in order to be compliant with the university rules. And sometimes that can kind of slow down the process. Many times we're in a situation where our science is funded, and that adds another layer of kind of complexity and bureaucracy and time to our work. And so what means that whenever we're pursuing scientific discoveries, it means that we can't always just kind of jump right in. We have to kind of go through a few additional hoops. Some other things that kind of slow down the process and make it a little bit more challenging, but important, are ethical considerations. Certainly in psychology, when we're studying human behavior, as well as animals, we have a responsibility to go through university approvals. We have the Institutional Review Board for any kind of science that we do on humans, and as well as a separate institutional review board for animal research. And this is an important part. I don't want to necessarily convey that it shouldn't be done. It's just an added layer, right, to the process. And so that can, contribute to some delays, just some extensions of the process overall. Here's a personal challenge that I run into considerably, and that is recruitment. So when I go off to do scientific studies, my field, as you heard, is industrial organizational psychology. And this is the blending of business and psychology. So my ideal sample of individuals that I want to study are people who are working in the workplace. And so in order to study individuals in the workplace, I often have to partner with organizations and gain what we call organizational entree. And so that usually takes time, developing relationships and a trust with the organization, and eventually access to the employees within the organization. Because inevitably they have their own, responsibilities and priorities that, so science and research that I'm pursuing is kind of an add-on and an addition. And so that just also adds to the complexities. It also means that sometimes as researchers, we might make some compromises when we can't necessarily gain access to the perfect sample. And so I was talking, in fact, to some of my students today about the fact that sometimes psychology gets a bad rap for doing too many studies on the psychology on the college student sophomore, and it's because, yes, they're a convenient sample, and we sometimes actually facilitate that by requiring students to participate in studies as a part of their Psychology 107 Introduction to Psychology course. But nevertheless, and this just gives us an opportunity to test out ideas initially, but we certainly have a responsibility to go beyond that, to make sure that our results do generalize beyond a college sample of individuals. And so just a few other thoughts about some of the challenges that we face in doing good psychological science. Ultimately, we want to strive for good quality data. And that means that we have to be rigorous in our methods. It means we have to take time to clean our data. It just adds another layer of importance to that process. And it also means we spend a fair amount of time designing our studies, picking out the measures, and analyzing the data. And so all of these things are considerable. The publication cycle adds to the layer of the process as well. Some of you I'm sure have personal experiences with this. I know when I was early in my career, it was surprising to me how long it takes to take an idea that you might have and a research question you want to test and take it all the way through to the end of the publication cycle, when you then go through the process of multiple iterations and the review process and revise and resubmit or reject and start over and all of that stuff. So just again, extends out before we can finally get our results out there. We also sometimes run into some challenges in terms of communicating our results. So as scientists, we have our very specific kind of technical ways of wanting to communicate the results, but sometimes we also want to also get that information out to journalists and to individuals who can get the information out to the broader public. And I'm sure many of my colleagues can appreciate that if you've ever had an interview with the media, sometimes the story gets a little bit distorted along the way. After all, journalists are in the business of putting out materials that are consumed by the public, and they're excited about hits or reads and so forth, and so they're going to want to make the story as sexy as possible, and it doesn't necessarily always align with the way that the science is necessarily presented. So there's a little bit of friction sometimes, I find, in terms of what we do in our studies and then ultimately how it's conveyed to the public, and that just a little bit of a disconnect. The last two things I'll just touch on briefly. I think that in many ways, our science is enhanced when we work together across disciplines. But I also find that that can be challenging in and of itself. I do a fair amount of work with my colleagues in the engineering department, but I find that we don't always speak the same language, and we have different words for different concepts. And so it takes a while for us to kind of come together and get on the same page before we can move forward in terms of contributing to the ultimate science that we're trying to pursue. And so it has its pluses and minuses nevertheless. And finally, a certain challenge that we have been experiencing here in psychology has been something that's been referred to as the replication crisis. And this is just going back to the fundamental issue within science, we do replicate. That's part of the scientific process, right? To test how robust our findings. And recently, some researchers have shown that there's been a number of studies that have been published in the psychology journals that cannot be replicated. And so it's definitely put a spin on us reconsidering some of the previous findings that we've had in the past, and trying to figure out why is it that we may not be able to replicate those results. And there's a lot of different reasons that have contributed to that, and that I won't necessarily have time to get into at this point, but it does certainly shine a little bit of a dimmer light on our scientific process when we can't show that replication. But nevertheless, it is part of the process. So we have a responsibility to pursue it and explain it and try to figure out which findings are robust enough to be replicated. So I think I'll pause there and let someone else take over.
Right. Well, thank you very much for the presentation, which I think provides both a wonderful context for the session as a whole and some specifics to challenges in psychological science. And so why don't we now turn this over to Dr. Rathore for his presentation.
Okay, thank you, Barbara. Thanks for the introduction, and thank you to all the A-STEP students for organizing this town hall, which gives us the opportunity to talk to people who are not in our area, and it's a great time to talk about this because GMOs are always in the news and many times not for good reasons. But so I want to take this opportunity to allay some of the fears people have and some of the questions they have. So here we go. Let's see. Okay, share. Can everyone see this? Okay, good. So I'm going to talk about one of my projects on cottonseed, and it's genetically modified cottonseed, and I'll tell you all about it. But before I do that, I would like to deal with some of the myths about GMOs, or sometimes we call them transgenics or genetically engineered plants, and what the reality is. Now, a lot of people worry about GMOs because they think it's unnatural. We are messing with the DNA and mixing DNA between different species. Well, it so happens that nature itself messes with itself. Certainly the DNA mutations occur naturally in all organisms. Even big chunks of DNA move around in the genome. And so that's nothing that we do is, which is totally crazy. And one other example I would like to give you is the bread wheat. So we eat bread every day. Now, nature produced this wheat by combining 3 different species. So here you go, you have this wheat that is just a product of 3 different species mixed up. Now, man created this species called triticale, and it's triticale is a wheat and rye hybrid. And we have been, it's mostly in Europe, they've been eating this for about 100 years. The other thing that I would like to point out is this bacterium called agrobacterium genes, which we use widely for making our GMOs. Now we are finding that many, especially the broadleaf plants, have genes from this agrobacterium. So nature has already done what we do in the lab. Okay, so that's the point. The other point I want to make is that we have been using mutation breedings, which actually means we apply chemicals or gamma radiations to create mutations, and then in whatever crop we are dealing with, and then that creates a lot of mutations. We then try to find the desirable mutation by screening lots of progeny from that. And so, and it's in just about everything that we eat. There are probably over 1,000 varieties that have been mutated one way or the other, and we eat and consume them without any question or any issues or concerns. Now, the other myth is GMOs are unsafe, approved with little scrutiny. At least here in the US, we've had 25 years of history of safe use of GMOs, and GMOs undergo intense scrutiny by the regulators. And in the US, we have USDA, FDA, and EPA to deal with. And another myth is that only large companies create GMOs and they do it for profit. Well, not always. There are some products that are available now and being used and consumed that have been created either by a university or a very small company. So papaya is one example created by at Cornell University. Arctic Apple is a small little company in Canada. The other myth is GMOs only help large farmers. Again, that's not necessarily true. For example, small farmers in India and China have benefited from BT cotton. It's one kind of GMO that fights the insects. And so for every dollar the farmer in developing country invests in GM seeds, he or she recoups $4. It's a little bit less in the developed countries. So I just wanted to talk about that first. Then I want to just show you just one example. And again, this is from my lab. And this is very easy to understand because it's you can see what I'm talking about. So this is with regards to cotton plants. So here is a cotton plant right in the top panel. And you see these black dots on the leaf and every other organ. The black dots represent glands. Glands they actually synthesize and store a compound called gossypol, which is toxin. And this toxin protects the cotton plant from insects. So this is the normal cotton plant. Now, thousands of years ago, maybe even longer, nature created a mutant. So there are two genes that control the formation of these black colored dots or the glands. In nature, and this cotton was found growing in Arizona. Nature created this mutant and it knocked out both the genes. So the two genes got mutated, and as you can see, there are no glands at all. Now in our lab, this is the middle panel here. You can see that this is a cotton plant with no glands at all. We created that using this CRISPR-Cas9 technology and did it in about 2 years. But we did this in a very precise manner. We knew what we were doing, where we are going, and what gene we want to mutate. So that's just one example of what's possible. So now I'm going to talk to you about this project we have in the lab. This is with regards to elimination of gossypol, which is the toxin in those dark colored glands that I just showed you, only from the seed. And the reason for doing that is to make this cotton seed available as a protein source for humanity. And some of the slides that I'm going to show you, I hope will convince you why this is such an important project. So here is a table that shows cotton production worldwide and the top 4 countries and some of the countries here in Africa. Now, and here is in this column, we have export numbers. So India, USA, Pakistan being the number, or China, sorry, China, India, USA, and Pakistan being number 1, 2, 3, and 4 top producers. These African countries are much smaller producers, but look at their export. They more or less export all the cotton they produce. And in fact, it's a big time export income earner for these countries. Gold has just become number 1 export income earner for a couple of countries, but it used to be cotton before that, but still cotton is number 2. For Benin, it's still number 1. So it's a very important product for them. Now look at where these countries stand in terms of global hunger index. So this was published about in 2019. It's 119 countries. India, that is number 2 producer of cotton, stands at 102nd place. Pakistan at 94th place. And some of these other countries are no better off. So there is a lot of hunger, a lot of malnutrition in these countries. And one of the main reasons for malnutrition is shortage of protein in their diets. A lot of people depend on they just survive basically eating rice or maize. And some people even just basically rely on eating cassava or yam. And this is the amount of protein these crops have, very low protein. So they're hungry for protein. And so we thought it'll be a worthwhile project to do, which is to deal with this cottonseed. Now, let me show you some numbers. So in 2014, we produced about 26 million tons of cotton fiber. At the same time, we produced about 47 million tons of cotton seed. Cotton seed has about 23% protein. And so what that means is that it has about 10 million tons of protein. Now, a human being requires about 50 grams of protein per day to survive. Now, if we can utilize all the cottonseed protein directly for human nutrition, we can meet the protein requirements of 500 million people. So that's a big number. And right now, we cannot utilize that protein for human nutrition because of the presence of this toxic gossypol, which is present in the seed and all the other parts of the plant. By the way, this little hill you see is a cottonseed pile right outside Lubbock. And if you want to know how big that is, here is a pickup truck right there. So you can have some idea as to how much cottonseed is produced. Now, as I said, cotton seed contains about 22–23% protein. It's a good quality protein. We cannot consume it, human beings, as well as we cannot give it to some of the monogastric animals, which are the single stomach animals like pigs and chicken and so on, because of the presence of gossypol in the seed glands. So if you take a cotton seed and slice it, you're going to see this dark color glands, and gossypol is manufactured and stored within these glands. As I said, gossypol is toxic to non-ruminants and it damages heart and liver. It's right now just given to cows or cattle. These ruminants can handle gossypol up to certain levels, so they are okay. And here is the structure of gossypol. So what is the current use of cottonseed? Well, we do extract cottonseed oil, but during the processing, gossypol is removed, and after that it's safe for us to eat. But the rest of the meal, which is high in protein, is fed to the cows, basically. Sometimes we feed the whole cottonseed to the cows. But all these other animals here, we cannot feed the cottonseed to these animals. Now, cows are not the most efficient animals in terms of converting crude protein into animal protein. So for example, it takes 20 pounds of feed protein to get 1 pound of beef protein. We call that protein conversion ratio, or PCR. So for cows, PCR is 20. Now compare that with these other animals. Pigs, 5.7. Chicken, 4.7. So chicken is 5 times more efficient compared to the cows, okay? The most efficient way to get animal protein is to have eggs. So the PCR for eggs is 2.6. More and more of our fish is coming from aquaculture, salmon 4.6, tilapia 5.7, here is shrimp. So now these, as I said earlier, these glands and the gossypol and some other related compounds, they protect the cotton plant from insects and some diseases. So when I started out on this project in 1995, end of 95, I had this goal, and the goal was to reduce the level of gossypol in the seed from 10,000 parts per million to about 450 parts per million. Now, why 450 parts per million? FDA considers anything safe, even for human consumption, if the levels of gossypol are below 450 parts per million. But we want to maintain gossypol and other related terpenoids in the rest of the plant so that it can protect itself from insects. So that was our goal. This slide just shows the various steps that go into making gossypol. So one thing to remember here is that cotton seed and the flower petals, the glands contain only gossypol. All the green parts of the plant contain gossypol and these other related compounds, but they all come from the same biosynthetic pathway. So we thought, okay, what if we block this step here? Can we knock and do so in a seed-specific manner? Can we eliminate gossypol from the seed without affecting the levels of this gossypol in other parts of the plant? And that's exactly what we did. We used this RNAi silencing. We silenced this gene in a seed-specific manner, and the results are shown right here. So here are two lines, and here are the seeds, 50 seeds in each case, and these are the gossypol levels. Normal, non-GMO seeds, these are the gossypol levels. So the gossypol level, so gone way down in these RNAi seeds. And we call this trait ultra-low gossypol cotton seed, or ULGCS for short. We took these plants to the field. Again, in the field, we see that the level of gossypol in these GMO lines are much lower compared to the non-GMO line. Over 3 years, we did this study. So this means that this trait is stable under field conditions. So far, so good. So we have eliminated mostly the gossypol in the seed. What about rest of the plant? Okay, so, and this is the organs that insects like to chew on. So we looked at the terminal bud, the floral bud, bracts tissue, petal, and so on. And the red bar again is the non-GMO, and the two green bars are the GMO that we created. And as I told you, that the green parts of the cotton plant contain not just gossypol, but other compounds which came from the same pathway. And none of those compounds are reduced in the GMO plants, meaning that this trait that we have engineered is completely seed specific. Okay, so the plant is still able to protect itself against insects. So that we did the science in 2006, published it. But if we want to take, because it's GMO, we want to take it all the way to commercialization, and so the farmers can grow and benefit from it, we had to go through a lot of other work. And this is what we did. We had to do a lot of other work, generated lots of lines again. We looked at where the gene was. We looked at all the biochemistry, inheritance. We carried out agronomic performance trial in 3 different states, 8 different trials over 2 years. We looked at the seed and made sure that protein levels was the same, oil level was the same. We looked at the fiber to make sure the fiber quality is the same. And we combined all that data and submitted our petition to USDA and FDA in 2017. We got approval, or we got this particular line got deregulated by USDA in 2018, and FDA gave us approval just last year in October. And by the way, we are calling it, since I'm Texas A&M, we call it TAM 66274, this particular line that got deregulated. So here we are. So the gossypol levels have been reduced from about 10,000 parts per million to 300 parts per million. And FDA considers it safe even for human consumption. But rest of the plant still contains normal levels of gossypol to protect itself against pests. Okay, so what can we do with this now? So we can now go ahead and take this cottonseed and feed it to the chicken, pigs, fish. People can use it directly. You can, put it in your tortilla flour and make tortillas or bread, and you can get more protein that way. Here are a couple of slides I'm going to just share with you to show you how much demand for feed there is in the world. And this is, these numbers are for broiler production in 3 different countries, plus this is worldwide. Now look at India and China.
Excuse me, I think we've gone a few minutes over time.
Okay,
so
this is my
summarize.
One more slide and I'll be done. All right.
Perfect.
Okay, so this is broiler production in India. The blue line is gone up by 51-fold. In China, it has gone up by 29-fold from 61 to 2017. And I'll skip that. This is about the egg production. And same thing with the aquaculture. More and more of our fish is coming from aquaculture. So we can use this cottonseed as a feed for these animals. So I will just kind of stop here, and I can show you some of the slides later on. These are some of the people who are involved in the project. So thank you very much, and I'll stop right here.
Thank you very much. That's just sort of an amazing story of research and its application and its potential to do huge amount of good in the world. I think we have a few minutes for questions and answers before our break, and so let me open this up for questions and answers. And I don't know if you'd like to make Aaron or someone else host now. We're still seeing your screen, and thank you. And so let's hear if we have maybe a question or two before we break. And feel free either to ask the question aloud, or if anybody wants to type a question into chat, I would be glad to read it. Yes, someone has a question for Dr. Payne. As scientific process has undergone scrutiny, there are trained scientists and engineers who have questioned whether science and scientific process is as objective as we think it is. Do you have any response to such remarks?
Well, I don't know that I can speak as much with regard to the perspective on engineers, but what I can say is that in all of the studies that that I do and that I read about in psychology, we make a very concerted effort to explain in detail about our measures. And so to me, that's our primary opportunity to be as objective as possible. And so because when I think about objective, I think about objective being the same for everyone, right? Rather than subjective. And so it's same for everyone, at least in terms of how we approach a given study. We do have a standardized protocol in terms of what people experience, and the metrics that we're using, we're administering them the same for everyone overall. So ideally, that also facilitates our ability to do replication. Inherent in the study of humans, though, there's definitely an element of subjectivity, no doubt. And I do want to acknowledge that probably where maybe one particular thing that might be a part of your question is the issue of the fact that we as scientists are human beings ourselves, and inevitably we bring our own biases and our own lens to our studies in the first place. And maybe one way to kind of combat that in some ways, though, is to have collaborators. And so we provide some checks and balances for each other, and try to avoid letting those biases and our lenses kind of influence how we approach our science.
Thank you very much. And there's a question from Dr. Cooper Smith, and I think this will be our last question before our break. And that is, what has been the reaction to the availability of the GMO low gossypol cottonseed in potential markets, both by immediate users, for example, chicken breeders, and ultimate users, for example, people eating the chicken?
Yeah, that's a really good question. I wish I had a good answer right now. The reason for that is that we've just had this deregulation and the FDA approval. And the next step is to try to see if one of these big corporations who actually are, biotechnology generators, they will pick it up and put that into their varieties that the farmers want to grow. So right now, that's what we are hoping for. But we've had many, many inquiries from other countries. For example, India, they asked us for these seeds, and also we just had recently, just a couple of days ago, inquiry from Argentina. They want this seed. But now we're kind of working out some arrangements as to how to share it with our international partners. So that's in the process. So it'll be some time before we are at the level of answering that particular question.
Yeah, interesting question. I could see that one as perhaps even something that both of our two speakers could work together on, because there is the psychology aspect and the GMO aspect. Sure. I think we are now, I think we're up to our break time now. And so why don't we have a 10-minute break as ASTEP has planned. I have 6:46 on my computer. So 6:56, we'll come back and I will condense my welcoming remarks then, and we'll be right on time. So have a good break and see you in 10 minutes.
Thank you.
Thank you, doctors. Those were really good talks.
Yeah, thank you both. Super talks.
Sorry, Barbara, I went over a little time, but.
Oh, no, that's okay. It worked out fine. It just so interesting.
But I even cut back on so many of my slides, and I think it's just a, you know, not an easy subject to cover in just, you know, few slides and all that.
No, absolutely. I mean, it's what 25 years of work and trying to get into 15 minutes.
Right.
No, this worked out fine.
Yes.
And everybody, if you're here, there was just a question about whether people will have more chance to ask questions to all the speakers. And yes, it's at the end, we will have a session that will be open to ask all speakers questions.
Yes, okay. Yeah, I'll be here as long as it takes. So.
Thank you, we really appreciate it.
Dr. Gastel, your Zoom background's lovely. What's it a photo of?
This is Texas wildflowers, bluebonnets, and Indian paintbrush. And this was taken on the Texas A&M campus near the—
Oh, it's on our campus?
Yeah, they're the gardens on West Campus.
Gotcha.
Near the agriculture buildings. In the spring, they have beautiful wildflowers. And I took a picture of the wildflowers, but I thought this composition of this picture isn't very good because it's just a field of wildflowers. There's no real focus. But then I realized it would be perfect for a Zoom background. So.
Oh, it's lovely. And I didn't realize you taking the photo. It looks gorgeous.
Yeah, so that was.
One more incredible skill on your resume.
It's not as good as a picture that my husband took. He took a better one in that area, but that had, you know, more of a focus and more composition, so it doesn't work as a background. So.
No, I think this one's lovely. Wow, I didn't realize our garden's right here on campus.
Yeah, it's a lovely
place
to take a walk. And I mean, even that's one, you know, I'm not on campus a whole lot now, you know, with you know, sheltering in place, but I do go over and take, you know, at times where I figure there will not be many people over in the gardens, I'll take a walk there.
No, it's lovely. Well.
And should we get started again, or should I wait about 1 more minute?
Go ahead and get started, right? Because I think folks will be interested in the big Q&A at the end too, it sounds like.
Okay, good. Yeah, so why don't, you know, there may still be people taking a little bit of a break, but it has been 10 minutes. And so why don't we get started? And again, I'm Barbara Gastel, I'm the moderator, and I have been asked to reintroduce our speakers in case some people came late and were not, don't know the speaker. So this time I'll just be quite brief, and I see we do have all our speakers here now. As mentioned, Stephanie Payne, Dr. Stephanie Payne, who was our first speaker, is a professor of industrial and organizational psychology in the Department of Psychological and Brain Sciences. Our second speaker, who was just talking about GMO cotton, Dr. Keerti Rathore, professor in Department of Soil and Crop Sciences. Our next speaker will be Dr. Charles Long, professor in the Department of Veterinary Physiology and Pharmacology. And then our last speaker today, whom I had introduced earlier, is Dr. Mark Holtzapple, professor of chemical engineering. And so without further ado, why don't we go to our next speaker, Dr. Chuck Long. And I've had the pleasure of having a glance at his slides and know he has some really interesting things to report. So let me turn this over to you now.
Mute myself. Hey. Thank you very much, Dr. Gastel. I appreciate that introduction, and I appreciate everybody showing up here tonight. You know, these types of forums are really interesting for me because it's an opportunity for me to see how much my scientific colleagues and I think alike. And so you're going to see a bit of a theme on certain topics here tonight that I think really just accentuates how important we really believe that some of this communication really is. So with that, I'll start somewhere with exactly where Keerti left off. You know, one of the things that we discuss a lot and we deal with a lot are common myths and misconceptions about what it is that we even do. And I think that his list was a little more extensive than mine, but we must keep in mind that, you know, our intentional modification of the DNA in our food is not new or novel, and it certainly is not dangerous. We've been doing this in different ways for hundreds of years. And so this concept that somehow we're fooling around with nature is just simply not a reason to move forward with these kind of technologies. Genetically engineered animals and recombinant DNA are not harmful. We know that because we have decades worth of experience now working with these, and there's no indications that they are harmful to any animals or humans. we tend to focus on the unintended effects or the
you have to like
and those the conception out there is that these are very common and they're serious, and it's actually the opposite. We get asked a lot because of our work with animals and livestock in particular about animal suffering. And I can assure you that there is not a single colleague of mine that would do an experiment that would intentionally create harm or suffering to any animal. We care very much for these animals, and we use them in our research, but they're treated with the utmost respect and certainly never made to suffer. And finally, you know, there's a strong misconception in the community that, and especially in the health conscious community, that organic and non-GMO food is actually healthier for you. And that is also absolutely false. There's no data whatsoever to support that. So that's the kind of background.
Let's see. I think you have muted yourself.
Thank you. You guys got you got me unmuted, but I think I'm back. So this is the background that we have to really present our talks and how we're going to communicate with the public, and that is incredibly important. Science communication is absolutely crucial for us, and both the scientists and the public really have to keep an open mind and approach this in the spirit of exchanging ideas and not in a more adversarial context. We certainly don't want to get into the same types of discussions that we see our politicians in every day. So we really got to do our best as scientists to communicate. And this is just a list of stuff that I do. I know my colleagues are doing the same thing out there, but I encourage those colleagues that don't regularly talk to the press to do so. Dr. Payne mentioned earlier that there are some risks with that, and that's true. But I have to tell you that if we don't try, then those myths and misconceptions are going to just get stronger as we go along. The new technologies that Keerti had mentioned about CRISPR-Cas9 and some of the approaches that we now have to modify the genome are really creating an ability for us to solve some very old problems. The difficulty is that in addition to these, our ability to solve problems, we have a perception that it's going to create these new fears, and Hollywood works against a genetic engineer more than anyone. And so every time we get a new technology, a new scientific breakthrough, seems to me like we get a movie that creates that in or restructures that breakthrough into a fearful thing. And I think what's important for us to communicate and to remember is that there's a lot of things that we should be very scared about in the United States. And we are living through an incredibly difficult year. And the pandemic is one thing that we have to be concerned about. But if you just take basic causes of death in human population every year, things like heart disease and cancer and respiratory disease, these are things that we really should be concerned about. And we can use technologies to cure and to treat illnesses like this. But what's worse than that really is what's facing the world population. And again, my predecessors tonight talked about this and how important it is for us to be thinking worldwide and about what the world faces in terms of a hunger crisis. And this, as of about 4 p.m. yesterday, were the number of people that have already died from hunger and hunger-related diseases around the world. That's a 9.7 million number as of yesterday. And that number will certainly hit top 10 million by the end of the year. So we have a pandemic that's causing us to be very concerned, but we seem to almost ignore the everyday loss of life due to unavailability of simple food products. I use this slide a lot because I think it's important to put it in context. Hunger is real and it is lethal. There's 1 billion hungry people around the world every day. That's about 1 in 7. We have heard about diseases like malaria and AIDS and tuberculosis, things like that that ravage underdeveloped countries around the world. And hunger kills more than those diseases combined. And the reality is that hunger is also very intricately linked to poverty, obviously. And so, you know, when we talk about livestock in developing countries, livestock are the key in many of these countries to wealth. And so if we want to make people have enough money to have a successful life and to feed themselves, then we need to have animals that can survive and are productive. And the population around the world shows no sign of slowing down. The current pandemic aside, we're going to stop that within the next few months, it looks like, and we're going to continue on a trajectory like this. And so we're going to have to slow this down. I know the next speaker is going to touch on this a little bit, and I hate to steal his thunder, but he's going to thunder it as well, I hope. And that is, we're dealing with a very changing world. The climate is changing. That change is real, and it will be lethal. So we're going to see a lot of the more difficult things. We've already seen one of the most productive hurricane season on record already. You're going to have a higher increase in droughts, devastating storms. The sea levels will rise, and there will be intense water shortages around the world. So what does this mean for an animal agriculture guy like me? We need to feed more people with less land. And the reality is that we are steadily losing the amount of farmable land per capita, per person on the world. The food demands are increasing because of because of population increase. Animal feed demands, as Keerti mentioned, are increasing dramatically. And as we go to a green economy, the need to produce biofuels is going to increase as well. So here's the question I ask some of my students. You know, how do you sustainably feed 9 billion people in about 25 years with less resources in an adverse environment? Sorry, 30 years in an adverse environment. And we could do it the same way we've always done it, which is basically waiting around for random mutations to occur in the offspring. And when those random mutations produce an animal with a better phenotype, with more production capacity, then we can select those and breed them to other animals that have similar phenotypes or production traits, and slowly do that over generations. And that's what allowed us to change an auroch into the modern cattle types like Holsteins or Angus or anything else you might see out there. But that process takes time, and the classical genetic selection technologies have made steady progress, but there are limits to that. You cannot select for a trait that does not exist in a population. It's simply impossible to select for a trait that doesn't currently exist. It has to occur randomly over time. Testing herds to discover individuals that are resistant to certain diseases is simply not practical. We're not going to release a disease like, say, foot and mouth disease in the United States to identify which animals have natural resistance. That would be catastrophic, right? So it's just not feasible for us to do that. And then finally, breeding to introduce these desirable traits can lead to the development of deleterious traits, meaning that sometimes as we're putting all of our selection pressure in one side of things, other parts of the animal tend to deteriorate. And ultimately, these things are slow, random, and they're limited to traits that already exist in the population. So we really have to come up with alternative methodologies to get to our goals in a reasonable amount of time. So that begs the question, you know, how do we use genetic engineering to solve some of these very serious problems? And we're all about trying to make animals more efficient and to prevent them from dying of needless disease. And so this is just one example of how you might make animals that are more efficient. Essentially, if you take your meat breeds and produce more meat per animal, then you could do so on less land and less production acreage. And this is just an example of a naturally occurring mutation called the myostatin mutation in a breed of cattle called Belgian Blue. And these animals have an extreme muscling phenotype that many breeders around the world would like to see in their breed. We don't use Belgian Blue cattle because they have lots of problems with dystocia, which means that they have trouble calving, and they have feet and leg problems from years of breeding for a single trait. They have created different problems with their skeleton. But we could do precision breeding and basically take the myostatin trait from these animals and place it into other breeds that don't have those other detrimental traits. And this is a project we've worked on in our lab, and it works very well to basically increase the amount of meat per animal. And I want to remind everybody, again, Keerti mentioned this, and I think it's important to remember, cattle eat grass, right? So if we can make these kind of cattle grow like this on eating nothing but grass, we don't have to feed them human food in order to get them to their slaughter weights. And so that's really what we want to do. I know I don't eat grass. I know some people sometimes smoke grass, but that's what we want to do, is make our cows eat something that we don't want and still have the same production efficiencies. We also want to make our animals resistant to pathogens. So diseases kill millions and millions of livestock around the world every year. And sometimes, especially in underdeveloped countries where vaccination strategies aren't in place, the diseases that sweep through these countries and kill livestock can be economically devastating for families. And when they get into poverty, then they can no longer feed themselves, and we create this really terrible cycle of hunger because of these diseases. So people can, we have done many projects have been done in the world to alter the way the viruses enter the cells, or to prevent the bacterial toxins from binding to the animal cells, and allow these animals essentially to be resistant to these different pathogens. We can also correct mutations in their genome. So sometimes, as I mentioned, there's genetic diseases that pop up. And this is an example of a project we did with a horse that had a genetic disease that was lethal when it was in a homozygous condition. And we can use skin cells from horses like this. We can correct the genetic disease using the CRISPR-Cas9 technologies. And then we can use these cells in cloning experiments. So we can clone those cells, and we can produce animals then that are an exact replica of the original, except it doesn't have the genetic mutation. And so this is a way, not only in companion animals like horses, but also in livestock, to eliminate genetic diseases from otherwise incredibly high value animals. And ultimately, as I mentioned, the environment is changing, and we have to be able to produce this huge amount of protein, huge amount of animal products in a very environmentally responsible way. And so this is a project that I really like to talk about. This was a project that was done many years ago in Canada, and they named this little pig EnviroPig because it was able to digest phytates. And phytates are at high levels in things that pigs eat, like corn and soybeans. And the animals normally cannot break down the phytate, and they excrete it in their urine and their feces. And this phosphorus that's in their feces and urine is a tremendous pollutant in our water systems and in our farming areas. And so we really have to find a way to decrease the amount of phosphorus that these animals are excreting. So they developed this pig that had an enzyme called phytase that was secreted in the salivary gland. And this pig had a 65% reduction in the excreted phosphates. But it never saw the light of day because of the anti-GMO movement in Canada. And this is the slogan they used. Why is the EnviroPig so full of, you know, stuff? And they begged the regulatory agencies in Canada, and they got enough people to sign petitions that the Canadian government stopped the project, and the EnviroPigs were all euthanized. So, and the project stopped. So this is just a great example of a project that had incredible potential, but was shut down because the anti-GMO movement had a very loud voice. And those are just a few examples of the things that have already been done in the world. And this is a lot more, a list of those gene editing innovations that we've done, my colleagues around the world have done to improve the performance of livestock production systems. We now have pigs that are—
See, we're a couple minutes over, so maybe summarize.
All right, will do. So we have pigs that are resistant, chickens that have resistance to disease, cattle, same way. We have the environmental improvements, as I talked about, with decreased phosphate pollution. These are all projects that have been done. There's cattle that are more heat tolerant, that have a hornless trait, so we don't have to go through the painful procedure of dehorning our cattle. We can have increased milk quantity and quality with genetic engineering. And that requires us to have a risk-based assessment of these foods in the regulatory chain. And I like to use this as well, and this is just simply a phrase that I think applies to all of our regulatory agencies. Common sense is so rare there that it should be classified as a superpower. We simply have to have an ethical, risk-based assessment of our technology, or we're going to have a situation where we will have some very difficult decisions to make in the future. The ethical position for doing nothing is much worse, I think, than the ethical position of trying to defend a technology that typically is being talked against out of fear of the unknown. And I always tell my students, you know, in this case, doing nothing is not an option. And I'll wrap up with this. I think this is a good way to let all the young people and even the older ones out there that, you know, we really, the people that present these talks here typically really love what we do. And if you do that and choose a job you love, you'll certainly never work a day of your life. So with that, I'll stop and be ready for questions later.
Thank you so much. Wonderful quote to end with, and a wonderfully quotable presentation. I can see why journalists interview you a lot. So thank you very much. And we will now move to our last speaker and then open it up for questions for those two speakers and then for everybody. And as mentioned, our last speaker is Dr. Mark Holtzapple, and he is going to be talking about climate change. So let me turn this over to you. Thank you.
You're muted, sir.
Can you see the presentation on the screen?
Yes, you're good to go.
Great. So my job is to address one of the largest technical challenges faced by humankind. And that's a pretty bold statement. We're actually involved in what I call the grand experiment. And the idea is, what happens to planet Earth when we dump a bunch of carbon dioxide into the atmosphere? And you see here, going back to 1850, how much tonnage of CO2 has been emitted every year. And you can see that this curve is growing exponentially. And as you would expect, as you dump more CO2 into the atmosphere, the concentration of CO2 increases. This is on Mauna Loa, an extinct volcano up in Hawaii. And you can see, starting back in 1958, there's less than 320 parts per million, and now we're well over 400 parts per million in the CO2 concentration. Now, going back to roughly 1860, you can see that the global temperature has been rising. And if you plot those two data sets together, the CO2 concentration in white and the global temperature in blue, you see that there's a correlation between these two data sets. Now, there's a famous quote in statistics that correlation does not mean causation. So we have to look at it in a little bit more depth. And so to do that, we use what are called global climate models. These are very sophisticated. They're based on first principles such as conservation of energy, mass, and momentum. All kinds of data are fed into those models. And what they do is they divide up the atmosphere and the oceans into a bunch of cubes, basically, where energy is flowing in and out of these cubes. And they model what's happening to the Earth. And so these are 4 models. And you can see that when you compare the models to themselves, the models are agreeing with each other. And when you compare the models to the data, which are shown as these black lines, we see good agreement with the data. So we have reason to believe that these models are reasonable representations of what's actually happening on the planet. Now, for me, this is what I call the smoking gun. Why is it that I take every opportunity possible to talk about climate change? If we take those models and we include all of the phenomenon, the natural variations, for example, in solar output, and all of the human-caused phenomenon, such as emissions of CO2, when we compare the observations, which are in black, and the models, which are kind of in this brownish color, you see that it agrees. So the models do a good job of describing the data. But if we run the model where we remove all the human factors, what we see is that prior to 1960, the models do a good job of describing what's going on, but after 1960, the models no longer agree. There's a divergence. And basically what we're saying is that after 1960, the anthropogenic CO2 starts becoming important. So what is it about the year 1960? Well, if we go back to that graph that I showed earlier, this is the emissions, the annual emissions of CO2. The area under the curve is the cumulative emissions since essentially the Industrial Revolution. And you can see the area under this curve is actually rather small. But after 1960, you see that the area under the curve is quite large. So what we're saying is that prior to 1960, the human impact on climate was negligible. We couldn't distinguish it from the noise. But after 1960, the human impact has been significant enough to actually start moving the climate. Now, if we take these models and we project into the future, what you see here, this is the actual data, and the blue curve is what the models say. Now, of course, it's hard to know exactly what's going to happen in the future, so there's an error band there. But if we use the dark blue as the best estimate of what's going to happen, what we can say is that the United Nations says we shouldn't have more than 2-degree warming above pre-industrial levels. So this line right here, the dashed line, is pre-industrial levels. And then here is the 2 degrees warming. And so when is it that the projection is will pass that threshold? It's roughly the year 2039, which is about 19 years from now. So it's really not that long that we are going to experience a warming of about 2 degrees Celsius above pre-industrial levels. And I don't really see anything that we can do in the next 19 years to bend that curve significantly. So this is something that we should expect by this time, 2039, thereabouts, to experience this 2-degree warming. Now we possibly can do some things that will bend the curve later, but so far we're not. There isn't anything that we're doing at an appropriate scale that's going to bend that curve. So the current trajectory is shown in this blue, and we haven't really done anything to change that trajectory. If we look at the 10 hottest years on record, 2016 was an El Nino year. That was the hottest. Last year was the second hottest. And you can see that these warmest years are all in recent time. This is, to me, quite dramatic. In 2016, that El Nino year, I found this graph, which is the temperature anomaly. That is, they compare to this base period. Between 1951 and 1980, the temperature was fairly flat and well known. And so what we're doing is comparing the temperature now to that temperature. And you can see up in the Arctic that it was about 11 degrees Celsius warmer than what it was in this reference period. Now that's just 2016, which, as I mentioned, was the hottest year on record, an El Nino year. So maybe that was unusual, but the reality is it wasn't unusual. In 2018, you see that the Arctic was now 12.3 degrees Celsius warmer than usual. And then in February of 2019, that was repeated in these regions right here. And then this year, you can see vast portions of Russia and Europe actually were almost 12 degrees Celsius warmer than usual. So you know, we often like to think of global warming as something that might happen in the future, but really it's something that is happening now, and you can see that in these data. And so what can we do to solve these problems? One is the use of biofuels. So I'll be talking about the work that I'm doing. If we take biomass, and there's all kinds of biomass that can be used, we put it through our process and we can come out with transportation fuels that are essentially gasoline. And we use what I call the big mouth. There's a biological process, which I'll describe briefly, where the various components of biomass are all converted into carboxylate salts. It's a natural process. And then using chemistry, we can transform those salts into a wide variety of chemicals and fuels. And so the process looks like this. We take the biomass and treat it to make it digestible. Then we ferment it into organic acids that are extracted. And then we use chemistry, often incorporating hydrogen, to transform those raw materials, the carboxylic acids, into chemicals and fuels. And then the undigested residue can be gasified and turned into biochar, which allows us to take low quality land and make it into high quality land. Now the fermentation process is the same natural process that occurs in the rumen of a cow. And so what I'm trying to do here is actually scale up to industrial sizes the natural processes that occur in the rumen. Now, that chemistry that I talk about, we can either start from the acid itself or the salt of the acid. Each of these arrows represents a chemical step, often involving a catalyst, and in some cases involving the input of hydrogen. And in some cases, these products are hydrocarbon fuels. So almost everything that we're currently making from oil and natural gas, we can construct those products from these renewable resources. This photograph was taken in 2010, so it's about 10 years ago. These are the first liter quantities of hydrocarbons and alcohols that we made from waste materials, waste paper, chicken manure, and food scraps from Sabisa Dining Hall. If we look at the economics, they depend on the scale of our process, what you pay for the biomass, and how you source the hydrogen. But the gasoline, depending on what these assumptions are, is about $1.24 per gallon to $2.56 per gallon. All of these prices we have experienced in our lifetime, and the economies didn't collapse. So these are prices that we can afford to pay. We've been working on this process since 1991. In the year 2000, we built a pilot plant, which is located on campus. And then in 2008, we started building a demonstration plant. Here you see the first engineering drawing. Here's the pit being dug, which is lined with concrete. And here you see the tanks that hold the fermentation broth. So this gives you a sense of scale. You can see a truck in the foreground, and these are the tanks that I just described. And so we're basically filling this pit with biomass. This is what it looks like on the inside. So imagine this whole interior volume filled with biomass, literally hundreds of tons of sorghum straw were put into this pit. And then the acids are in the water. We collect the acids in this sump right here. So this is a photograph of what the structure looks like on the outside. This is an actual photograph. The next photograph is an artist's concept of what it should look like, because what we're doing is scaling up the cow. And this is an aerial view of the facility. So there's that building that I was describing to you. There's also some fermenters. There's submerged fermenters over here. So you can see this is 90 feet in height, so it's pretty large. And this is the chemical plant that takes those organic acids and transforms them into useful products. We've actually started building the first commercial plant this year. They'll actually be making specialty chemicals that are high value. So what we're saying is that this is really an infinite cycle. We take our biomass and go through processing, ending up with gasoline that we burn in our car. The CO2 that comes out of the tailpipe is actually fixed by photosynthesis. And so now we have an infinite cycle where the carbon goes round and round and round. And as long as the sun is shining, we can drive this process. And we can also even use natural gas as our source of hydrogen. The CO2 that comes from that can be put back in the ground so that it doesn't accumulate in the atmosphere. The next topic I'd like to touch on is high efficiency engine, which is illustrated here. This is what we call the star rotor engine. It's the same thermodynamic cycle as used in a jet engine. So what I'm trying to do is put a jet engine in everybody's car. What you see on the left is the compressor. There's a heat exchanger here. The fuel is burned in the combustor. So it's very, very hot, high pressure. And then that hot, high pressure gas drives this expander. It comes out still pretty hot. We extract out the heat from that and send off into the world a relatively cool gas. So this is a remarkably efficient machine. Here you see it packaged in a kind of a three-dimensional geometry. There's the combustor, there's the heat exchanger, this is the expander, and this is the compressor. So these are efficiencies of conventional engines, gasoline engines, gas turbines, and diesels. Each of these data points represents an actual engine that you can buy on the market. This is a star rotor engine operating at 1500 Kelvin. So you can see it's significantly more efficient than conventional engines. And if we raise the temperature to 2000 Kelvin, it's even more efficient. So one of our challenges as an engineer is to drive up the operating temperature. If we were to grow sorghum as the feedstock, this is planted as a seed in March. This photograph was taken in November. So you can see that in basically one growing season, it grows this tall. So if we were to have conventional cars powered by sorghum, they get about 30 miles per gallon. Driving cross country would require about 93 gallons of gasoline. And if we planted this amount of area in sorghum, we could supply all of US gasoline needs. However, if we put a star rotor engine into a conventional car, we'll now get about 90 miles per gallon, and our fuel requirements drop to 31 gallons, and our land area shrinks. If we put a star rotor engine into a more efficient car body, this is the Precept, a concept car from General Motors. Now a cross-country trip would require only 20 gallons. And if everybody drove cars that looked like a Precept, that amount of land area would be sufficient. And if we go to the Volkswagen XL, we could get 180 miles per gallon, driving down our fuel requirements, reducing our area. And if we go to even more efficient car, 10 gallons would be sufficient to drive cross country. And if everybody were willing to drive cars like this, that amount of land area could supply all of our gasoline needs. So the main conclusion I'm trying to to communicate to you is that if we want to address global warming, we don't have to go back to the horse and buggy days. We can, in my view, we can maintain the quality of life that we've all love and are not going to give up, but we can do it in a way that doesn't cause global warming. And with that, I'd like to conclude, and I think we're going to go now to a question and answer session. So I'll be quiet and look forward to addressing questions that might come up from the audience. Thank you for your attention.
Yeah, thank you very much for another fascinating presentation. I think one theme that I've seen go through, I think all 4 presentations, is that there are problems out there, there are creative solutions, but the creative solutions sometimes do take time and some patience, and it's been exciting to have a couple of sort of longitudinal views from among these presentations. I think now we will open it up, first of all, to a couple of questions for Dr. Long and Dr. Holtzapple, and then we will, I will be returning the session to the ASTEP members, who will moderate a general discussion and have some closing comments. So let me open this up for questions, and feel free either to ask questions orally or to type messages in chat, and I'll be glad to read them. And meanwhile, do we have any questions from our organizers from ASTEP?
Hi, doctors. Well, I thank you both so much for a fantastic, for 2 fantastic presentations. I don't mean to steal the thunder from folks in the group chat, though. So it looks like there are 2 questions in there. I can always follow up with you guys. So Dr. Gastel, do you want to share those, or do you want to?
Oh, sure. Yes. Yeah. We now have a couple of questions in our chat in case people aren't following chat. The first one is for Dr. Holtzapple. Would it be possible to use biomass directly as an energy source through micro and nano materials? And let's see, this just went off my screen. Would direct use of biomass energy source be more efficient?
Okay, so you have to be clear about the form of energy. If you're going to, let's say, make electricity, it definitely would be more efficient to burn the biomass directly. When you go through all this conversion technology, there's always loss. You can't avoid it. The reason for going through all this conversion technology is to have the fuel as a liquid that can go into your car. You can't put a straw into your car and have it work. It has to be a liquid, and preferably gasoline, because that's what our cars are designed to use. There's also a discussion about, let's say, nanomaterials. There actually are people looking at cellulose as a nanomaterial. It has some amazing strength properties. So people are exploiting that right now. But my personal view is that if you're going to convert biomass into something useful, if you start talking about nanomaterials, it usually becomes mega expensive. But what I try to do is very simple technologies that more or less align with nature, so that you don't spend a lot of money. So that to be economical, it needs to be very simple and robust. But certainly, for high performance materials that money is no object, then I would say yes, nanomaterials are perfectly fine.
Thank you. Thank you very much. I see we have a link that Dr. Gaddy has added, and Dr. Gaddy would
No, it was an act. Please delete it. That was trying and paste a link, a question, Dr. Long.
Okay, if you'd like to just add a question to Dr. Long, that would.
Into the red that it had.
Yeah, I've done the wrong cut and paste ones.
It was actually my question was about what kind of resources might be out there for people that want to support the whole idea of genetically modified organisms for animals. I mean, there's, you know, the anti-animal folks. Is there anything that the public can do, we can do to help that effort, to support it? I mean, you got any ideas on that?
Yeah, thanks, Dr. Gaddy. The best website I've found is one called the Genetic Literacy Project. And I actually meant to put that at the end of my talk today, but I just forgot. So the Genetic Literacy Project is essentially, it's a non-profit organization that is set up to try to just put the truth out there. They don't have an agenda necessarily to be anti-GMO or anything else. They just want to try to dispel the myths that are being created by the anti-GMO movement. And so the Genetic Literacy Project is a great place to start. And from there, you can find lots of different links and different ways to support the use of genetic engineering.
Thank you. And we have another question also for you, Dr. Long. Someone asking, are there any current efforts to bring back the EnviroPigs?
Yes. Yes, there are. And they're being done in China. In fact, most of the genetic engineering projects right now are being done in China because the Chinese government is very supportive of that effort. They know that it's an absolute requirement for them to maintain food production in their country. So they have done more work in genetic engineering in the last 10 years than all the rest of us combined, I think. It is remarkable the pace that the Chinese scientists are on in terms of producing animals with disease resistance, enhanced production traits, milk that has improved properties for human consumption, and especially things that can improve the environmental aspects. So that project has been kind of restarted.
Thank you. Interesting. In a moment, I think I am going to turn this back to our A-STEP moderators, and then there'll be the chance for to ask questions of all of our speakers. But as you know, sort of closing thought, as I close my part of this, I really want to thank the A-STEP organizers and all the speakers for just really great use of our evening. I was a little bit leery coming into this. I was thinking Friday the 13th, maybe it's going to be unlucky, what's going to go wrong with this. But then I realized this is second Friday, and I think this was even better than first Friday in downtown Bryan. So thank you all, and let me now turn this over to our A-STEP organizers.
Dr. Gastel, thank you so much for moderating the night. And of course, a huge thank you to all of our speakers as well. Thanks to everyone for being here. Like, this event is not possible without you. And, you know, we do it for you guys, and it's been a joy to have everyone here. So as a small thank you from our team to all of you, I just wanted to make sure that if you haven't registered for your free meal pickup at the Village Cafe in downtown Bryan, you know, go ahead. The registration is still open, so go ahead and make sure you're signed up for that. I'm going to go ahead and post the pickup date for each of the meals in the chat here. So if you signed up for the pasta, your pickup date is any time from whenever the Village Cafe is open, essentially, on Wednesday of this coming week. And then if you're here for the vegan paella, that's on Thursday, and then the red wine chicken's on Friday. But beyond that, so we'll feed our mouths, and then right now we're feeding our brains. So I won't keep you guys for too long. I'd like to open up the floor back up to all four of our speakers. So folks, if you have any more integrative questions, or if you want to double back to some of our past speakers, this is your chance. So again, I encourage you, feel free to unmute yourselves and ask your questions, chat, and we'll try and keep the ball rolling for discussion, okay? Thank you all so much again.
Erin, there was a question in the comment box that was addressed to me.
Yes.
With regards to if everyone adopts our cottonseed, that's going to create problem because it can become susceptible to whatever disease or, you know, all this stuff. That's not the way things work. So what we have done is just introduce just one trait into our cotton plant. And this variety that we have transformed or engineered is a old variety. Nobody uses that anymore. But breeders can take this trait and transfer it to the current varieties that the farmers want to grow in their field. And those varieties that farmers are growing currently are adapted for different regions. They have, you know, disease resistance, drought tolerance a little bit, whatever traits they have, they already have it. So ours will be an added trait to whatever is there in particular variety. And this is going to differ, all these varieties are going to differ from region to region and country to country and so on.
Thank you, Dr. Rathore. It looks like there's another question for you also in the chat box. It looks like this attendee wants to ask, can you tell us more about the process of getting the Holtzapple, I'm going to mispronounce that, Holtzapple cotton seeds?
Yeah,
yes,
well, I had that slide there. Once you have kind of got the science down, one of the things that they do require is, and say you want to go through USDA and FDA, luckily EPA was not involved in our case. We had to generate hundreds of lines and try to find a line that has our gene and has done the job of silencing. But we need to find lines that are stable over generations. We had to find lines that have just one copy integrated of this RNAi cassette into their genome. We had to make sure that they performed well agronomically. There was no problem with the fiber quality or the seed quality, with the exception of Holtzapple. So we had to do all that. Now, if you're more interested into this, you can go, you can just email to me and I will send you a link to APHIS website where you can download this, all the 400 pages of the data that went into the evaluation.
Sounds good. We'll be sure to pass on any resources. So again, if anyone has any questions they'd like to send to our speakers afterwards, please feel free to contact A-Step and we can make sure that we put you in contact with the appropriate expert here. So are there any other questions? I think, oh, there's a question for Dr. Long. Is there more resistance now to GMO animals with increased interest in lab-grown meat and plant-based meat substitutes?
This is a good question, and I get this one from time to time. The idea that we're going to be able to just utilize meat substitutes instead of actual meat is out there, and it's growing. But the reality is that the scale up of those processes is going to be very expensive, and it's going to take a long time. I can't quite understand, just from a personal perspective, why someone that prefers to have a vegetarian-based diet would even care to have their vegetarian-based diet look like a hamburger. But that's just me. I would just soon have the vegetables the way they are and have my steak on the side. Is that changing the views of GMO? I think if anything, it's enhancing the anti-GMO movement, because it gives them the idea that there are alternatives to using animals. You have to remember that a large majority of the anti-GMO movement is also confounded with the animal welfare movement. And so because those two things are very closely linked, we get get activism in both sides. And so if you are anti-GMO, typically you may also have the viewpoint of you shouldn't be using animals at all for anything. And so I think it's increasing, if anything.
That's a fascinating take. Thank you, Dr. Long. Dr. Holtzapple, there's a question for you right now. How does the biomass and star rotor research and adoptability get impacted by the commercial use of electric vehicles?
That's a wonderful question. You're basically saying you're going to substitute electricity and have it replace gasoline. And that's increasingly happening. There are some challenges. Batteries are actually quite expensive. And there's a lot of environmental impacts from mining the lithium and cobalt that goes into the batteries. Only a few percent of automobiles now are all electric. It's just a tiny fraction, but it is growing. My personal belief is that the cars in the future will be hybrids, where we plug in for our travel, let's say our daily commute, but we still have a fuel-powered engine that can take us long distances for a trip. So for example, the Tesla is perhaps the most famous electric car. If you want to go on a long trip across country, every few hours you have to stop and maybe take half an hour to an hour to get it to charge, where it only takes 5 minutes to fill up with gasoline. So I think, if you have a commuter car in your family that's all electric, that's tolerable. But if you want to have a car that's general purpose, I think it needs to have the ability to use fuel. And then there's some applications where no matter what, batteries can't substitute. So airplanes, for example, you're not going to have an international airplane that's powered by batteries. That's just not possible. So there will be a substitution of electricity for fuel, absolutely, and I'm in favor of it. I think it's a good idea. But I think in the long run, there's going to be some new balance where we still have to have liquid transportation fuels. That's just unavoidable. Also things like heavy equipment, tractors, like farmers' tractors, earth-moving equipment. You're just not going to do that with batteries. So there's always going to be a role for fuels.
Thank you, Dr. Holtzapple. It looks like, there's another question for you. Are there plans for the biomass processing plants to be close to crop fields as to reduce transport costs and degradation of sugars?
That's another great question. My vision is that you don't move the biomass very far. That, for example, there might be a cooperative that has the process. Think of Producers Co-op here in town, perhaps having a facility where farmers can bring their residues and have it transported. The carboxylic acids have about 88% of the energy content of gasoline. So my vision is that we transform the biomass into carboxylic acids at the biomass site, and then transport and consolidate those acids at a refinery and upgrade them and turn them into fuels and chemicals so that we get that economy of scale. So my vision is that literally thousands and thousands of facilities across the country would be turning biomass into acids, but maybe only a few dozens of facilities would upgrade the acids into final products.
Excellent. Dr. Payne, we have a question for you. So during your presentation, you talked about the role of trust in communicating science. What advice do you give to early researchers and students like us who are maybe just starting out in our fields?
That's a tough one. I think that I'm intrigued at this whole event. I wasn't knowledgeable about A-Step, so I think it's really awesome that you guys are organizing something like this and having an opportunity to witness how researchers are presenting their research to others. And I think that as graduate students, you should just be sponges. So just take advantage of every opportunity you have right now to learn and expose yourself to lots of different speaking styles in terms of kind of being able to communicate what we do. I think I mention this to my students all the time. You may go to a talk and you may not necessarily be real intrigued with the content, but you can still take away something from it. So, and it certainly that, the ability to communicate about our science to one another, and to build the trust to lead to people understanding what we're doing can come from those experiences.
Thank you so much for sharing that insight. Dr. Long, you've had a lovely line. Actually, you've had incredible one-liners all night, but another one I'd like to share to everyone in the chat in case folks don't see it. Never compromise your integrity. You'll never get it back. Fantastic.
Yeah, that's the, you know, if you want to build trust with the people that you're communicating with, you better start with the truth. And it's very tempting for a student to walk into his or her professor's office and really want to give them good news when it's not. We prefer the truth, just so you know.
Aaron, I would like to add one more website, or actually there are two that Dr. Long mentioned. One was the Genetic Literacy Project. You can get a lot of good information about all sorts of GMOs and everything. FDA has just kind of started another website. So if you just Google FDA and feed your mind, you'll get to that. And I think it has a lot of good information, basic information about GMOs, both, I believe, crops as well as animals. So that, and the third one is, it's ISAAA, or stands for International Service for Acquisition of Agri-Biotech Applications. So that also has a lot of good information on all kinds of GMOs.
Thank you so much. I'll definitely throw those resources into the group chat as well, so that way everyone has access. And then just as a reminder for all our viewers at home, this talk will be recorded, so we can definitely go back and access these resources too. Looks like there's a question for Dr. Payne. Given the current use of social media and sensational articles, what are some tactics one can use to communicate with a stranger that the stranger's sources are flawed, other than saying they're flat out wrong and or lecturing them on the subject?
That's challenging for sure. And as somebody who's compelled by science and data, that's all I need to be convinced if I'm mistaken. I think there's something to be said about not over lecturing, right? And being angry. I think we have to kind of keep our emotions separate and try to stick to the data. I think I'm persuaded by the number of people out there that are on social media that are presenting positive things too, and informed, information. So maybe it's, I'm hoping that eventually those sources will outnumber the other ones, but I also tend to see the world as rosy. Maybe others on the panel will have ideas on this.
No, those were excellent insights, and I think you're right. I mean, I tend to take an optimistic outlook. How do our other presenters feel?
Well, I think you have to find common ground. I think most people can agree with Mark's talk that things are getting difficult and we have to find innovative solutions. I think everybody can agree that with Keerti and I that hunger is an issue that we're going to have to deal with, and we need to find new and inventive ways to solve those problems. The way we do it is different, and we may have disagreements on the way we go about it. But I think that if you can find the common ground between two people that are in stark disagreement about how to proceed, and you don't have to look very far other than the last election about this to get a lesson in this, but there's a lot of commonality, and we can agree that we need to solve the problem. We may disagree on how.
That's incredible, and I think very timely advice. You're right. In light of recent politicization of a lot of issues, I think the advice of finding common ground and remembering, hey, we're all on the same team at the end of the day, should help a lot. So thank you for that. Alrighty, folks. Well, it's 8 p.m. We won't keep you out too late on a Friday night. Does anyone else have any last minute comments or questions? Going once, going twice. All right. Well, thank you all so much. Again, thank you so much to our speakers. You guys were all incredible. What a wonderful panel. I really enjoyed this evening. And of course, thank you to Dr. Gastel as well. And then thanks to all of you. You know, this wouldn't have been possible without your viewership either. So, all right, Aggies, have a good weekend. Thanks, everyone.